Hi, my name is Blythe Marshman. I'm a research scientist here at the Shellfish Health Lab at Bodega Marine Laboratory, and today, in honor of National Oyster Day, I'm going to show you how to shuck an oyster. So what you're going to need is either a glove to offer your hand a little bit of protection, or a towel. You're going to need an oyster knife. This is also called a shucking knife. It has a nice long blade, but the end is very dull. And you're going to need yourself an oyster. So these are Pacific oysters. These are the most commonly farmed oysters we have on the West Coast. The species name is Crassistrea gigas. You'll notice that they have a cup side and a flat side. So what you're going to want to do is either with the towel, you'll want to put the cup side in your hand. For the demonstration today, I'm actually going to use the glove. And you're going to want to put it in your hand with the pointy end facing towards you. This is what's called the hinge, and this is how we're going to actually get into the oyster first. The oyster is only attached to its shell at two parts, at the top here and at the bottom, using a muscle that's called the adductor muscle. So what we're going to want to do is we're going to want to get into the shell via the hinge here, and we're going to want to go along the top to break that connection with the adductor muscle. You want to use pretty steady force, but not excessive force. You want to be very careful with this. And you want to make sure that it goes along the oyster as opposed to in it, because that's how dangerous things happen. So I'm going to put my hand down here for a little stability. Get into this hinge a little bit. This is the part that can sometimes take a bit of effort. And you'll feel it. You get in that hinge right there. So I'm in right now, and then you're going to twist it. And you're going to break it right there. And then once you're into that hinge, you're just going to use that top part to go right along the flat edge. And the top part of the shell will come right off like that. If you're going to use these for barbecuing or eating, get any little bits of shell that came out. And then I'm going to want to break the connection with the adductor muscle on the other side. I'm just going to do that. There we go. And now that oyster is nice and loose, ready to be popped into the mouth of a hungry individual or put on the barbecue. In our lab, this is the point where actually we would take some samples of the tissue to look for abnormalities with this oyster. So we would take a sample of the gill, which is right here, maybe part of the adductor muscle. Their stomach is right here, that's called the digestive gland, and we put it in these little cassettes. And once we have that tissue in the cassette, we're going to preserve it and then take thin sections of the tissue to create a histology slide. And we're going to look at that under the microscope to see if we see any pathogens or any evidence of disease. We can also take little bits of the tissue and put it into a tube, and we can extract the DNA from that tissue. And by doing so, we can look for the DNA of pathogens that we might find. Particular pathogens that we're always looking for on the West Coast are Banamia, Denman Island disease, and OSHV. I have been working with oysters here for about 14 years. I think they're really cool individuals. They can filter 5 liters of water a day, so they're really good at clearing up bays. They're an incredibly sustainable source of protein. And one question I frequently get as somebody who studies disease in oysters is, do I eat oysters? And I do. My favorite is actually the Kumamoto. And I always encourage people, if you haven't tried an oyster, go to your local farm, go to your local seafood market, give it a go. And if you are a fan of oysters, maybe try a different species. So thank you for joining me today, and happy shucking.